Hey, what's up guys? Welcome to a new video tutorial from my everyday science. In this video we are going to talk about recombinant DNA technology. Basically we are going to look at the introductory aspects, and then in our next videos we are going to talk about the whole process into detail. So since it's an introductory lecture, I'm going to make sure it's very simple and also precise. So we may want to look at the definition of a recombinant DNA technology. And now it is the joining of different DNA molecules from different organisms and then inserting it into a host organism to produce new genetic combinations that are of value to science. So for our recombinant DNA technology, all we want is to clone a gene. Okay, after cloning this gene, then we introduce it into an organism so that it will show the traits that we desire. Okay, so if you scrutinize our definition here, it tells you the whole process of DNA technology, the recombinant DNA technology. So we are going to be needing genes from different organisms. Now what we do is that we bring these genes together by an enzyme known as the ligase. This one is going to facilitate the joining of these two genes. So we will be looking at these terms into detail in our next videos. So we bind these new genes together and then we replicate them in an organism known as our host, which we mostly use bacteria for this work. And then it produces new genetic combinations, and now we can use these new genetic combinations in the various fields of science for practical work. So simply put, our recombinant DNA technology is the intentional modification of an organism's genome, for short. Now, gene cloning. We may have heard of clones in several aspects, and it's basically the same everywhere. Clones just means copies. Okay, so if you say gene cloning, it actually means we are making copies of a gene. Now, this is to replicate a fragment of DNA placed in an organism so that there is sufficient to analyze or use mostly in protein production. So for our recombinant DNA technology, we would isolate a gene of interest. If we take an organism's genome, we identify our gene of interest, which is the gene that we are actually interested in. Now we pick this gene, and what we do is that we isolate the gene by means of a restriction enzyme. Now when we isolate these genes, we need to replicate these genes to multiply them so that we have sufficient amount. And this is what is known as our gene cloning. So simply put, we are just producing copies of a gene or a DNA. Now let's look at our requirement for the recombinant DNA technology process. So for our process, we are going to require a restriction enzyme, and as well we will require a vector, which in this case we would be using the plasmid DNA vector. Now let's analyze our restriction enzymes to get to know what they actually do. So when I was talking about gene cloning, I made mention of the fact that we would need to isolate a particular gene of interest from an organism's genome. And this restriction enzyme is what actually helps us to isolate that gene of interest. So when we pick an organism's genome, there are several other genes and then DNA molecules which we are not interested in. But the ones we are interested in is what we cut from the entire genome, and this is done by the help of our restriction enzymes. So as their function suggests, they are called molecular seizers, and they are produced mainly by some bacteria, and what they do is that they are for protection, for defense against external forces in the environment. So they cleave DNA molecules by breaking the phosphodiester bonds between adjacent nucleotides. We are well aware of the fact that successive nucleotides bond together with one another through phosphodiester bonds. So what happens when these bonds are breaking? Now, when these bonds are broken, we do not no longer have DNA, but rather we have nucleotides. Okay, so when we take this out of the organism's genome, then what do we insert it into our host, which is our vector, which we are going to talk about very soon. So they cut sequences of bases at sites called the restriction sites. So these restriction enzymes do their work by locating the sites with which they are supposed to cut, and these sites are called our restriction sites. How are they able to identify these restriction sites in the genome of an organism? Now, this is what they do. The restriction sites are all palindromes. Now, if you say palindromes, that means the sequence have the property of reading the same forward as it does backwards. Okay, so when you read the sequence from right to left, it gives you the same as reading it from left to right. Okay, so we can have an example such as race car. When reading race car from our R, which is in red, it reads race car, and from our R, which is also in blue, it reads race car. So race car can now be considered as a palindrome because from both sides it reads the same. We are going to look at how a DNA molecule or a site is said to be palindrome in our next video, which we are going to actually scrutinize the whole process and know what it actually entails. So now on to our vector or our plasmid DNA. Now when we isolate this gene of interest, we cannot use this gene of interest because it's not enough, okay, or it's not sufficient enough to help us in our experiment. So what we do is that we use these vectors, and this is what the vectors actually do. So they are molecules used as vehicles to artificially carry different genetic materials into a host cell where it can replicate. So this is what we do. When we get our restriction enzymes, we get a vector. So we cut our gene of interest and then we insert it into this vector, and then we now introduce this vector into our host organism, which most at times we use bacterium. So for our discussion today, we will use plasmid as our DNA vectors. So these plasmid DNA are smaller pieces of DNA which are found in the cytoplasm of bacterial cells. So we know the chromosomes of bacterial cells are located in their cytoplasm, as well as their plasmid DNA. So these plasmid DNA are circular forms which are also in the cytoplasm, and they can replicate independently of the chromosomes. So because of this, they are called extrachromosomal material or extrachromosomal DNA. So what we do to this plasmid DNA is that we use our restriction enzymes to also cut some sequences, and these sequences are also palindromes. So it's cut some sequences so that we fix our gene of interest into this vector, and then we can now safely introduce it into our host for replication. So let's now look at the steps in recombinant DNA technology. So first of all, we need to obtain the gene of interest, which is to isolate the gene of interest. So we pick an organism's DNA and then we locate our gene of interest. When we locate this gene of interest, what we do is that we apply restriction enzymes and then they cut these sites for us. So when we get these sites, then we get our vector. We cut the vectors also at their restriction sites, introduce this gene of interest into the vector, and then we insert it, insertion of desired DNA into the host cell for replication. Most of the times our host cells are bacterial cells. So we introduce these into the bacteria for replication. Now there are steps involved in these ones, and then we are going to look at all the different types with which we can insert a DNA into a host cell. So when this is done, what we do is that we get or we extract the product. So many a times when we introduce a DNA into a bacterial cell, it can produce the proteins that we want. So when it produces these proteins, we can simply extract the proteins for our experimental work, or we can get the DNA and then introduce them into other species for them to show the desired traits that we would want them to see. So this would bring us to screening the cells to identify which of them was cloned, because it's very difficult to find out if the steps that we carried out was really successful or it actually wasn't. So when screening these cells, we identify whether it was cloned or it was not cloned. Now this is the general steps in recombinant DNA technology, and in our next videos we are actually going to look at everything into details. Please don't forget to subscribe and then click the notification icon so that you get a notification whenever there is a new video.